Let's learn about today biotechnology. Okay, let's subscribe this channel for the more biology and geography sessions. So biotechnology is a fascinating field of science where we use living organisms or their components to develop products and technologies that improve the human life. It spans medicine, agriculture, environment and industry. So let's get started. The scientist in lab holding test tube with glowing liquid. Welcome to the world of biotechnology. So this visual introduces the field. It shows a scientist in modern lab ready to explore life at a molecular level. Biotechnology uses microorganisms, cells and DNA to produce useful products. DNA stores genetic information. Understanding its structure is fundamental to biotechnology because all genetic engineering revolves around manipulating DNA. Microbes like bacteria and yeast are vital in biotechnology. Microscopes help us study them, which is essential for microbial biotechnology, fermentation, and drug production. This illustrates lab experiments where scientists combine enzymes, substrates, and other molecules to understand biological reactions and produce new products. Bacteria can produce antibiotics, enzymes, and vitamins. Growing them in controlled lab conditions is a key step in industrial biotechnology. This introduces one of biotechnology's major achievements, producing human insulin using genetically engineered bacteria. Biotechnology improves crop yield and nutritional quality. GM crops resist pests, tolerate drought, or produce extra vitamins. This visual shows real-life applications. Farmers benefit from biotechnology through higher productivity and better quality crops. PCR, polymerase chain reaction, is a technique to amplify DNA. It is used in diagnostics, forensic science, and genetic research. Cloning is reproducing genetically identical organisms. Dolly, the first cloned mammal, demonstrates the potential and ethical considerations in animal biotechnology. Microbes convert sugar into alcohol or acids. This is the basis for making beer, wine, and biofuels. Bacteria are used to produce dairy products. This shows practical use in daily life. Modern vaccines are made using microbes or recombinant DNA. They protect humans from deadly diseases. DNA analysis helps identify individuals, solve crimes, and study genetic diversity. Gene guns are used to insert genes into plant cells. This is one method of creating GM crops. Bioreactors allow large-scale growth of microorganisms to produce enzymes, antibiotics, or biofuels. Plasmids are circular DNA molecules used to transfer genes between organisms. Restriction enzymes cut DNA at specific sites. This is crucial for recombinant DNA technology. Sticky end allow DNA fragments from different sources to join, forming recombinant DNA for gene cloning or protein production. This highlights the importance of recombinant DNA in producing medicines, enzymes, and GMOs. E. coli is widely used in labs because it grows fast and can be genetically modified to produce proteins like insulin. Lab safety is critical in biotechnology to prevent contamination and protect humans from harmful microbes. Plant tissue culture allows mass production of identical plants in vitro. This shows how crops like banana or sugarcane can be cloned for uniformity and disease resistance. Algae can produce biofuels and eco-friendly energy alternative. Antibiotics like penicillin are made by fungi and bacteria. This visual connects microbiology to medicine. GM crops can be engineered to produce more vitamins or resist diseases, improving the food security. Animals help scientists study diseases, test medicines, and develop therapies. Pipettes and lab instruments ensure accurate measurements for reproducible results. DNA sequencing decodes genetic information, helping understand diseases and evolution. Gene therapy treats genetic disorders by replacing defective genes with functional ones. Some bacteria can generate electricity, showing how biotechnology helps renewable energy. Microbial enzymes help produce eco-friendly plastics, reducing the pollution. Enzymes speed up reactions in detergents, food processing, and pharmaceuticals. GM crops reduce pesticide use and promoting the sustainable agriculture. Biopesticides from microbes target pests without harming the environment. Genetically engineered bacteria or yeast produce human insulin for diabetics. Microbes can break down pollutants, cleaning oil spills and toxic waste. Different experiments require different biosafety levels to prevent accidents. Modern vaccines are made via recombinant DNA, safer than traditional methods. Gel electrophoresis separates DNA fragments by size, useful in forensics and research. The Human Genome Project sequenced all human genes, revolutionizing medicine. Proteins can be modified to improve stability, activity, and therapeutic use. Genes can be inserted into bacterial plasmids to produce proteins like insulin. Stem cells can develop into any cell type, promising therapies for damaged tissues. Stem cells and growth factors help repair organs, a key application of biotechnology. Genetically modified fish grow faster or resist disease, improving aquaculture. Plants can be engineered to produce vaccines or therapeutic proteins. Bioinformatics uses computers to analyze genetic data essential for the research. Accurate records ensure experiments are reproducible and scientific conclusions are valid. Microbes are used in food, medicine, biofuel, and pollution control. Fermentation converts sugar into ethanol, an alternative energy source. Bioplastics reduce reliance on petroleum-based plastics. DNA cloning produces multiple copies of genes for study or industrial use. CRISPR allows precise gene editing, a revolutionary biotechnology tool. Safety measures prevent accidents while handling microbes or chemicals. Bacteria are engineered to mass produce proteins, enzymes, or vaccines. Visual emphasizes research and discovery in molecular biology. Biotechnology has evolved from brewing and cheese making to advanced gene editing. This final visual shows biotechnology global impact in medicine, agriculture, environment, and industry, highlighting its importance for humanity's future. So these all collective information, the journey of biotechnology from basic lab experiments to advanced medical and industrial applications. They cover all the DNAs, microbes, GMOs, recombinant DNA, vaccines, stem cells, biofuels, bioplastics, CRISPR, and global applications, providing a complete interactive learning guide. So for the more or next session for the biology, please subscribe this channel.